I recently did a new RNA extraction, and I wanted to give a quick visual of what this entails. The purpose of an RNA extraction is to extract all of the RNA from my frozen diatom cells. These diatom cells are from my experimental assays, which exposed cells to different environmental conditions and took cells at different time points. So far, I've just been extracting RNA from the first and last time points. I use this RNA to make complementary DNA, which I can then quantify using real-time PCR. The process starts when I take my frozen cells out of the -80 freezer and decide which samples I want to process. The majority of the RNA extraction happens in the fume hood because one of the stabilizing reagents I use smells pretty terrible. Because the first few steps take a while, I have to do them with the fume hood door mostly closed, dramatically increasing the finesse and agility required to do them. As you can see, working in the fume hood is pretty crowded and cramped, which is why I don't really like doing them. Once I'm all set up, I individually scrape the cells off their filters and wash them off of the buffer. Once I have all the cells possible, I transfer them to new tubes filled with beads that have been soaked overnight in buffer. This is the bulk of the work, and it takes me about an hour to do it properly. I then bring my filled tubes to a lab 2 doors down and load them onto what's called a bead beater. The bead beater shakes the tubes, and the beads inside the tubes help break up the cells. This process increases our RNA yield. After beating the cells, it's a matter of applying the aqueous supernatant to centrifuge filter tubes and removing the liquid. This leaves the RNA bound to the filters in the tubes, which I will then clean with a few rounds of buffering. This process kind of bites because it takes about 7 rounds of centrifuging, which times 8 samples is a lot of fiddling around. Once I'm finished, I have clean but delicate RNA that's ready to be made into DNA. To go from the experimental assay, to the RNA extraction, to the cDNA synthesis, to the real-time PCR process, it'd probably take me about 9 to 12 hours of straight lab work. To try it in one day sounds pretty difficult. Challenge accepted! But I might even try to do it one day, just to say that I did. This, however, might reduce the quality of my work, which would pretty much suck. So yeah, check out my other videos I've been doing recently. They're relevant to this topic, and I hope you enjoy them.